I'm Sushmita Roy. I'm an associate professor at the University of Wisconsin-Madison. I'm at the Biostatistics and Medical Informatics department, but I'm actually a computer scientist. I'm actually a computational biologist. So my group develops machine learning-based methods, applies machine learning-based methods to look at problems in gene regulation. And what I wanted to do today is talk about some of the problems that we are working on, and I'm hoping that it might give you some ideas and some. And please feel free to ask questions during the presentation if something is not clear. All right. All right, so let's get started. So the title of my talk is to come up with methods to look at cell type specific network inference. And when I was actually putting this talk together, I realized that actually this is a very generic problem. So I just adjusted my talk a little bit. It's actually context specific network inference. So let me start off with a very high level overview of what I mean by a regulatory network. And as many of you might know, a regulatory network or transcriptional regulatory network describes connections between regulatory proteins such as transcription factors that bind to the DNA of a gene. And my pointer is not pointing, but that's okay. Is there another pointer that works? That's okay. To drive the expression level of a gene. And when we think about a regulatory network, there are two components that are of interest. One is the structure of the network that specifies the wiring diagram of regulators to target genes. And the other is the relationship of how regulators actually drive the expression of a gene. Is it activating? Is it combinatorial? And so on and so forth. Another aspect of regulatory networks, as you might know, is that they are context specific. So networks change across different cell types, across different species, across different organisms and tissues and diseases. And what we would like to be able to do is to be able to identify what are the individual context specific networks. And then once we have this, we can try to compare them and identify what makes something more pathogenic versus not, and what defines differences across different cell types, and so on and so forth. But this is a hard problem, identifying these networks computationally as well as experimentally. And the reason why this is difficult is because there are many factors that determine whether a regulator is going to regulate the expression level of a gene. And here I'm listing a few of these examples, and some of you might have heard from the epigenomics focus group where about some of these things. But there is this notion of post-translational regulation that is controlled by signaling networks. So a protein can bind to the DNA but not actually activate the expression level of a gene unless it is actually phosphorylated or something. And then there is this notion of long-range gene regulation as well as chromatin accessibility that can complicate whether a particular regulator can regulate the expression of a gene. And all of these can be measured under different contexts. So in today's presentation, I want to talk about three approaches that we've used and others have used to try to get to inferring regulatory networks, context-specific networks. And the main difference between all of these different approaches is the number of samples that is needed and the type of data that is being used. So the first approach is conceptually perhaps the most simple approach of trying to infer a context-specific network, and that is based on this class of methods called expression-based network inference. So what is expression-based network inference? And I apologize for some aspects that you can't see, but at a very high vanilla level of what is expression-based network inference, it's basically taking gene expression data sets that are context-specific. So one way to infer a context-specific network is to collect context-specific gene expression data and feed it inside a network inference algorithm, and out comes a network. And there are a lot of different classes of algorithms that are out there that differ how they describe the relationship between regulators and target genes. So there are Boolean networks, there are differential equation-based models, and the ones that we've been working with are based on probabilistic graphical models. So people have actually applied this kind of an approach to infer a context-specific network, and here I'm listing some pretty high-profile papers that have done this kind of a thing, where they've inferred, say, the network for the Th17 cells or dendritic cells, which are special type of immune cells. But the issue with most of these approaches is that they've assumed that there are a large number of samples that are available for that cell line or cell type of interest, and that has been collected from the same lab, and most of them have used, with a few exceptions, they've used only gene expression data. So we started wondering whether we could actually deal with a situation where we may not have a single lab that is able to produce so many hundreds of different samples. So this was a project actually in collaboration with Randy Ashton, who is also in Wisconsin, and his lab is interested to study early human brain development, and he's using an in vitro system to look at very, very early on in humans how the early brain and the spinal cord develops. And in his lab, they've been able to measure gene expression profiles using RNA-seq for this very early part of development. And what we wanted to do with this data was ask this question: what regulatory networks control this early neural development? But the issue here was that this was only 20 time points, and with 20 time points, we are likely not able to infer a gene regulatory network of thousands of genes. So here is our approach, and this is really application of an approach called MERLIN+P, which I'll talk about in a few next slides, for this particular problem. And so the idea of our approach is conceptually simple. So what we are saying is, yes, we get a cell type specific expression data, which may not be a lot, and then we take another network which may belong to the system that is not exactly the same that we are looking at, and then we use this network inference algorithm that tries to refine the network by overlaying the cell type specific expression data. So what is MERLIN+P? And so MERLIN+P is a network inference algorithm that uses gene expression as input and some prior knowledge, and it tries to output a regulatory network that integrates both the gene expression and the prior information. And there are some of the highlights of this approach is that it is based on a class of models called dependency networks. So some of you might know about Bayesian networks. So dependency networks are actually a relaxation of Bayesian networks and are much easier to learn if you are willing to make some approximations, as in you are not so interested in computing the data likelihood. And this is an extension of a previous approach that we had developed several years ago, and where the goal of this approach is to predict regulators for not only individual genes but also groups of genes or gene modules. So briefly, what does a probabilistic graphical model representation of a regulatory network look like? Let's assume that it's too bad I really don't have a laser, but I'm going to use this mouse kind of a thing to represent where the pointer should be. So let's say you have an example promoter, and you have these two regulators, and we want to represent this as a regulatory network. Each of these nodes, HOT1 and SCO1, the regulator, and HSP12, they become random variables, and then we want to represent the relationship among the random variables using a graph structure, and in a probabilistic graphical model, we estimate some probabilistic functions. In the case of Bayesian network, they are conditional probability distributions. So what is nice about probabilistic graphical models in terms of representing a regulatory network is that both the questions of structure and function are naturally encapsulated. So the graph structure gets you what the wiring structure is, and the conditional distributions tell you how the regulators potentially regulate the expression of a gene. So that's what a PGM representation is. And so what did we do? We wanted to not only just use gene expression data, we wanted to use something more that would enable us to infer a network that is more biologically meaningful or more realistic. And so we used a prior-based framework. So the idea of this approach is to think of the thing that we want to infer, which is the graph, as a random variable, and we put it inside a Bayesian framework where what we would like to be able to do is estimate the posterior distribution of the graph given the data, which is proportional to the data likelihood and a model prior. And the idea of using a prior-based framework for network inference is that we use this additional information or additional knowledge that might be there as priors in the graph, and then that together with gene expression data infer a gene regulatory network. So just to talk about that in the context of a transcriptional regulatory network, different sources of prior information that might be available that can be informative for a regulatory network are things like measurements that come out from a ChIP-seq experiment, which tells you this transcription factor likely binds to the promoter of a gene, or a knockdown or knockout experiment, which says there's a significant change in expression given if you knock down the particular transcription factor, and so on. So those kinds of things can be used to describe this prior distribution on the graph, and then together with gene expression data, we want to get out we want to use both of these things to come up with what we believe is a relationship between a regulator and a target gene. So how might we go about describing this P of G? So what is G? G is basically, if you want to think of it as a random variable, it's actually a very high dimensional random variable, which has as many dimensions as there can be total number of edges. And so trying to describe a probability distribution over this very large random variable is difficult. And so what we've done and what many others have done in the field is to make some very strong independence assumptions, which is to assume that the P of G decomposes of a product of individual edges or individual pairs of variables. And then so we write it as a product of edges that are present in the graph and edges that are not present in the graph. And then all we need to do is describe the prior distribution, prior probability of a single edge. So how do we do that? We describe that as a logistic function, and this logistic function has a set of terms where the beta s's or the beta s is one term which is modeling how sparse or dense we want the network to be. Typically regulatory networks tend to be sparse, you pay a cost of adding an edge. And then the rest of the terms under the summation are basically terms that can be used to incorporate different types of prior knowledge. So just to come back to that example of these three sources of prior, so we have, say, chip, motif, and knockdown or knockout. We can write that as the sum of these three terms. So we have three terms for each of these different sources of prior. So we have this information for each of these edges. So, so I just wanted to tell you some of the key points of the MERLIN+P algorithm. So it's basically based on dependency networks, and the way we learn the graph structure given the data is based on a score-based approach where we're using a greedy search over the space of possible graphs. But this is much easier because it is based on a dependency network where the goal, where it just boils down to trying to find the regulators of each gene one at a time. And one of the things that we do is we feed this inside a stability selection framework. So what is a stability selection framework? It's similar to a bootstrap framework. The idea is to basically generate different subsamples of the data, and for every subsample get out a network and then get some kind of a consensus of the different networks. And then what this gives you is a ranking and edge strength for each of the edges in that one infers. All right. So that's MERLIN, and what it does is basically takes an expression data and some prior network and then gives the output a network. So the first thing we did was to ask the question, how well do we do? So we, even though most of our results are in mammalian systems, we start off with always yeast. Whenever we come up with a network inference algorithm, yeast is the best model system to first see whether the algorithm is working, because there are a lot of good data sets and a reasonable number of gold standards. So here what we did was we used a gene expression compendia and we used as prior sequence-specific motifs, and then as a gold standard we used chip. So how well do algorithms do when we actually apply this prior-based framework and compare to methods that don't use these priors? So here I'm listing a set of algorithms, and you see basically two sets of curves. These curves that are on the top over here are methods that make use of priors, and it includes MERLIN as well as another method from Richard Boneo's group, which that all use priors, and methods that use prior are able to do much better. And by better, I mean they're able to get much higher precision, and the precision recall basically looks much better than what you get if you didn't use prior. And this is what we see with methods that don't use prior. And so now what we are doing is we are trying to use this in mammalian systems where we are collecting publicly available gene expression data sets from Gene Expression Omnibus for a few select cell lines that people have used, and then we are trying to apply this and see if it helps. And what we see is for some of these cell lines that there is a significant advantage in terms of inferring the regulatory network if you are using prior-based information. Now, coming back to our original problem of trying to infer a neural stem cell or early hindbrain specific regulatory network, how do we use this approach? So what we did was the following. We looked in the Gene Expression Omnibus and asked for data sets that were relevant or similar to early hindbrain development, and we got those data sets. And then we used as prior a DNA's accessible network. So basically we took a motif network and then we just filtered out edges based on accessibility that came from the Roadmap project, which has studied some neural stem cells or neural progenitor cells, and we inferred, we fed that inside MERLIN and got out a network, and then we used that as a prior for together with the data that was generated in Randy's lab, and then got out another network which we call the neural stem cell specific network. So this was our approach, where the idea is to basically take a network that is somewhat relevant but not exactly, and then just refine it by applying this prior-based regulatory network inference algorithm, and then get out a final network. So what does our network look like? Here is the hairball picture of a regulatory network, and what you're seeing here are some of the regulators that I've just highlighted that have a lot of connections, and these are some statistics for people who are interested. There are about 6,000 edges, and if we rank the regulators based on the number of connections, we see several hawks and other regulators that have been known to be important for the neural stem cell development. And we use the network to try to prioritize what might be important regulators, and what we find is that several Hawks-related regulators that have been known to be important for patterning, as well as in this particular system, are ranked highly in the prioritization. So one of the things that we in our lab is very interested to do is to do validation. Of course, we are computational people. We don't really know how to do experiments, but we collaborate with other people, and so we wanted to do some experimental validation. And so what we did was we took some of these top regulators and then tried to validate some of these edges. So I'm just going to go quickly through some of these things. So this is the Hawks A5 centered regulatory network, where the edges are just color coded based on what we think are activating or repressive relationship. And this is another subnetwork that we inferred that was around the POW3F2 gene, which again was predicted to regulate these different target genes. So we wanted to validate these, and how do we go about validating it? So what one can do is try to do these siRNA experiments, where one can knock down the regulator, and then depending upon what we predict the expression, the edge weight, one can predict whether there is an upregulation or a downregulation. And this is being done in a very low throughput way, but if somebody has the firepower to do it in a high throughput way, I think that would be a huge benefit to the community who are interested in network inference. Anyway, so what we did was we took four regulators that we knocked down, and we validated these edges. And one of the nice things that we found was basically one edge that we were able to validate, and this was based on the POW3F2 gene. And so what I want you to focus on is basically ignore the green bars, just focus on the yellow bars and the blue and the two blue bars. The two blue bars are basically controls, and the yellow bar is what happens to the expression of this particular gene when you knock down POW3F2. So we had predicted that POW3F2 has an activating relationship to STMN2 and RFX4, and it represses these two. what we find is that when we knock down POW3F2, we see a significant downregulation of STMN2, and of course POW3F2 also has to be downregulated. So for the others, there is some change in expression, but we cannot say anything significant. But what was nice to see is this particular relationship, and the POW3F2 is a known neural regulator, and STMN2 is known to be involved in neural stem cell specification and neural development, but this relationship was not previously known. So just to summarize the first part of my talk, what I talked about was a prior-based framework to generally integrate gene expression data with other types of prior knowledge. And what we used was repurpose this approach to try to infer a cell-type specific network where we were leveraging publicly available data sets that were relevant to the system of our interest. And then we applied our MERLIN+P network inference algorithm to identify regulatory connections. And then with the help of our collaborators, we were able to validate a few of these connections. So the second part of my talk will talk about another approach, which is also simple to do, and that's based on skeleton-based network inference approaches. And the difference between skeleton-based network inference and what I talked about in the first part is that in skeleton-based approaches, you can't add new edges. You're basically just taking a skeleton network, and then you're identifying what edges are there. And so here is very simply, you have a set of you have a skeleton network which has a, we are assuming that it has a union of all edges that we care about, and then you have some context specific gene expression data, and you're basically overlaying the data onto your system of interest. So here is a picture. So we have our skeleton network, and we have 3 contexts, and the way one can try to identify what edges matter are we can do one of 2 things. One can do something really simple where one can compute some kind of a pairwise correlation or pairwise metric that says that these 2 edges should be present. It depends on the number of samples. If you have enough samples to be able to compute Pearson's correlation, you could just compute that, or you could do a differential expression analysis and say these 2 nodes are on, so I'll allow edges that are connected by 2 regulators that are both on, or 2 genes that are both on, and then outcomes a context-specific network. The thing that we did was something a little bit different, where instead of considering pairs of edges, we used something where we just took the graph structure and used that to serve as a skeleton on which we just solved a regression problem. There have been several approaches that have used something like the basic idea is to use something like this. So we wanted to use this kind of an approach for a system that we've been studying together with Rupa Sridharan, who's a biologist who's interested in cellular reprogramming. So cellular reprogramming is the process of turning a differentiated cell and turning it into a pluripotent cell. And so the cells that you get out are called induced pluripotent cells, and once you have this, you can try to differentiate them to different types of cell types. And so getting these types of cells and doing them efficiently is an important problem in regenerative medicine. The problem is that this is a very inefficient process, and it takes time, and the number of cells that actually can become pluripotent is quite small. And so in her lab, they've been using a variety of different genomic approaches to try to look at this problem, and many others are actually interested in this problem. And one of the things that they found recently was that if you apply a particular type of chemical combination called A2S, which is really a concoction of 3 chemicals, ascorbic acid, 2 chemical inhibitors, and another chemical which actually kind of represses a particular histone demethylase or histone methylase. If you do that, you are able to get a very high reprogramming efficiency. And then she wanted to characterize this, and she used single-cell RNA-seq data and then applied a bunch of different standard analyses, such as identifying trajectories and so on. And where we came in was we tried to answer this question of what makes A2S more efficient. So the way we did this was we started with our original A2S data, and the FBS is basically the older media. So she had RNA-seq data for A2S as well as FBS, and then we fed it inside MERLIN, and then we got out modules as well as a regulatory network. And in parallel, she measured a new single-cell RNA-seq data set where she measured the individual combinations, the dual combinations. So they are A2, which is basically the ascorbic acid, and the 2I, S2, and AS, basically the 2 different pairwise combinations, all of the 3 pairwise combinations at day 4 and day 6, which is basically different time points during the reprogramming process. And then what we did was we took the MERLIN modules and networks, and then we used that as a scaffold. And then for every gene, we basically re-inferred what the edge weights might be given these different gene expression data sets at each time point and cell type. So just to give you an example of what we got, here is the MERLIN inferred network, here's the hairball picture, and here are the modules that we get. And the different modules that were identified exhibited interesting trajectories. So these the columns are ordered based on the certain cell clusters, and also what they do going from the MEF state to the embryonic stem cell state. And looking at some of these modules, some of the things that we identified was so what makes A2S more efficient is that in the dual combination where you don't have all of the 3 components, some subnetworks, just especially those that are associated with the somatic state, that is the mouse embryonic fibroblast state, they don't seem to be downregulated. So in the AS type, we see this particular network that seems to have much stronger connections than what you see in the A2S state, which is towards the end. And then another module that we found was that in A2S, the embryonic stem cell regulatory network, which is composed of important regulators like NANOG and ESRRB tend to be much more strongly upregulated. So one of the things that we were able to say using this kind of an analysis was that in the A2S combination, the embryonic stem cell state is much more, the regulatory network that is needed for the embryonic stem cell state is much more strongly upregulated together with the downregulation of the somatic programs, whereas in these individual dual combinations, these different programs are not as strongly down or upregulated. All right, so that was the second part of my talk, and again, the idea of this approach is to take a skeleton network, which could be an expression-based network, and then overlay gene expression or context-specific expression, and then filter out edges that might be important for the context of interest. And the main difference here is that we didn't really add new edges, whereas in the first part, you can actually add edges because you're actually doing network inference, but you're doing a more constrained version of a network inference. And then we applied this to single-cell RNA-seq data, where we tried to use this kind of a framework to identify what parts of the network might be on or off, depending upon the particular treatment that was given. All right. So how am I doing on time? Like 15 minutes? Okay. All right. So this is the third and the last part of my talk, and here I'm going to talk about some very new work that we are doing, and that's based on multitask network learning. So one of the motivations of trying to use this kind of an approach is the following. So biological systems tend to be hierarchically related. So when we think about a cell lineage, we start with a starting cell types, let's say an undifferentiated cell type that gradually over time, different regulatory programs get activated, and what we see are different cell types that are of increasing specificity. And across evolution, the same thing happens where we have species related by a phylogenetic tree with a few differences that we don't have any observations of the internal node, but they're related by a tree, and there are networks that are associated with each of these different species. And what we would like to be able to do is use these kinds of relationships to try to infer these cell type or species-specific networks. And the opportunity is that now we are able to measure not only gene expression data, but also we are able to measure other types of data that are informative of what an edge can be. For example, we can measure accessibility, which can tell you from a single experiment, provided you have the motif information, whether a transcription factor actually binds to the particular DNA of a gene. And there's chromatin information. And what we would like to be able to do is try to use all of this information and then try to infer a regulatory network where we are trying to get the structure and the function. The challenge is that we typically don't have a lot of samples. So the previous approaches kind of relied on the presence of a sufficient number of samples. I mean, the skeleton-based network, not so much. It's much more flexible. But if you want to do network inference starting from gene expression data, you need a substantial number of samples. But I'm talking about a situation where you have, say, a few dozen cell types, where for each cell type you've measured, say, one mRNA profile and one ATAC-seq profile and one chromatin profile and so on. And you want to be able to infer a regulatory network. So what we've started to do is to develop an approach that I call dynamic regulatory module networks. And the idea of this approach is to relax. So we make an approximation. So instead of learning the regulatory program for individual genes, what we are saying is that let's group genes together into modules. And we are not the first to come up with this. People have used this kind of a module-based approach a lot of times. And and then try to predict what does the expression module, what specifies the expression module state. And another thing that we add is to leverage the fact that there is a lineage relationship that relates these different cell types. So before I get into the DRMN model, I want to tell you about the regulatory module network. And so the idea is, let's assume that we have a very simple cell type where there are only two expression states, high and low. So each of these little boxes correspond to a gene. So some genes are high, some genes are low. And what we would like to be able to do is predict the expression state as a function of their upstream regulatory program. And these programs are again defined by transcription factors that bind to the promoter of a gene, so basically cis-regulatory features. And so basically a regulatory module network is made up of the regulators and the different modules, and then how the different regulators describe the expression level of the gene. And the DRMN approach is basically trying to apply the RMN for each of these different cell types. And what we want to do is try to learn these regulatory module networks across all of the cell types simultaneously. So for every cell type, we have a graph structure, but here the graph structure is kind of connecting individual regulators to expression states instead of individual genes. And we have parameters that describe how the expression is specified. And in addition, we have something called transition matrices that specifies how the genes change their module relationships. All right, so that's the basic DRMN idea. Some things that one might wonder about is how do we actually share information in terms of learning the regulatory program simultaneously. There are two things that we are trying. Both of them are interesting and have interesting results. So one idea is to use something like a structure prior, where we are just trying to impose similarity on the structure of the edges, so what edges are present or absent. And the second approach is trying to use more of a regularized regression kind of an approach, where we are sharing information at the parameter level. Most of this work has been done using linear regression models. The advantage of the structure prior approach is that you can actually have much more complex models that don't have to be linear. So just to summarize, what does the DRMN approach do? It is based on a predictive model of expression states. In order to deal with the fact that there are not very many samples for a particular cell type, it works with the level of groups of genes that are in a particular expression state. It tries to integrate different types of chromatin data sets, accessibility information to try to predict the relationship between a regulator and expression state. And it does this by using a multitask learning framework, where each task is basically a cell type, and we are trying to infer the regulatory module network for each cell type. All right, and so we applied this approach to, again, Rupa's system, where she is looking at MEF cells going to pluripotent cells. And we used two types of data. One data set was when she was a postdoc in UCLA, actually. And she also some sequencing data that was more recently generated, which is a larger version of the data set that we had at the array level. And basically, the sequencing data also has attack-seq data, which we didn't have the array level. And to this, we add sequence-specific motifs. So how do we look at these regulatory features? We are essentially looking at just the promoter of a gene and then looking at the regulatory signal. So we are looking at the number of reads that are aligned to the promoter around the TSS of a gene. And the chromatin as a feature is simple, where we just look at the number of reads that are associated with a particular marker on the TSS. Motifs are easy also, you just look at which motifs are present. Attack-seq, just looking at attack-seq is just look at the number of reads that are accessible, number of reads that are lining up around the TSS. And then finally, we looked at another feature, and there are actually different ways in which one could model this. We are using a somewhat simple approach where we want motifs that are quantified by the attack signal. And so we just look at a motif and look at the attack reads that are aligned up around the motif. And we are using these all as predictive features to predict gene expression states. How do we evaluate how well we did? So remember, the tasks that we are trying to solve are trying to predict expression. And what you're seeing here is predicted versus actual expression levels, and for each of 5 modules that we inferred. And if you just look at global correlation, it will actually look very good. But what we would like to be able to do is say something at each module level. So for each module, we try to estimate the expression level, and then we take the average of the predicted versus actual. So we looked at the different features that we have, and this is on the one side we have array, and the other side we have sequencing, and as a function of different modules. And we are asking the question, how well can we predict expression? And in blue we have the what we can predict based on just chromatin state, and in yellow is what we can predict based on motifs. And what we see is that chromatin is a much better predictor of expression, which is not a surprise, and people have seen this many times. But when we add motifs, we are able to get an even better performance, and the signal is much better for array data than for the sequencing data set. Remember, this is all happening at the promoter. If you look at long range, maybe you'll get better predictive power. And the other thing that we found was that when we jointly learn when we use an approach like DRMN versus single task learning, which is the RMN approach, we see advantage in terms of doing this jointly across different cell types. So in red is what you see with DRMN, and in blue is what we see with RMN, and we see an advantage in trying to use this joint learning framework. All right, so DRMN as an input, it takes in the number of modules, and we are still working and analyzing this data, so I have some initial results. So what you're seeing here are the modules that we get from DRMN. So we go from the MEF cells to the ESC cells. In each of these cell types, we have inferred 7 modules, and what we are doing right now are interpreting these modules, and one way to interpret them is to look at what they're enriched for. So here is a module that is repressed in all of the different cell types, and we tested them for enrichment, and we find that some of them are actually associated with differential enrichment. And this was an interesting enrichment, which is TCF3, which is known to downregulate the embryonic stem cell program, and it's kind of enriched in the MEF cell type. And similarly, for the more induced module, we see some other regulators that exhibit some amount of differential enrichment. And one thing that we can do with the DRMN approach is, and which is perhaps more interesting, is to identify genes that actually go from different states over or different modules that go over during this process of reprogramming. So here is a trajectory that we've outlined, and here are some genes that go from the most repressed module in the MEF state and become induced in the embryonic stem cell. So these are I can identify several such gene sets, and then look for enrichment. And this was a particularly interesting gene set, which went from an induced state to a repressed state from the MEF to an induced state, and it was enriched for a particular regulator, the TCF3, that is known to be down-regulating embryonic stem cell programs. So just to summarize the third part of my talk, what I talked about was an approach called dynamic regulatory module networks. And the idea of this approach is to deal with very deep data set, as in like you have many different types of measurement, but you don't have many samples of these types of measurements. And we have several cell types for which we have this type of information, and we want to be able to say something about the relationship between regulators and gene expression programs. And in addition, if they are related by some kind of a lineage structure, we should be able to leverage this kind of information. So with that, I just want to summarize what I talked about today. I talked about this general problem of context-specific network inference, where the goal is to infer what the network might be for a particular context. And a context can be different diseases, different cell types, different species, or different environmental conditions. And I talked about three ways in which you could do this. The first approach was saying that let's try to use context-specific gene expression data, assuming there's lots of it, a substantial number, and then do network inference. The second approach was saying, maybe we don't have very many samples, so let's try to just overlay gene expression onto these kind of a skeleton network. And the third approach was trying to integrate many different types of data, but relax the requirement that you're learning these fine-grained models, but rather learn more coarse models where you're trying to predict the relationship between regulators and expression state rather than individual genes. With that, I just want to thank my collaborators and my students. And at the background is our building. And we actually have trees inside the building. So if you like that kind of stuff, I invite you to come to Madison. And I'm happy to take questions. Thank you.